My name is Gianni Zifarelli. I'm Senior Director of Business Development. I'm following some of the pharma collaborations we have with our partners. And it's a great pleasure to introduce you to our two speakers, Peter Bauer, Centogene Chief Medical and Genomic Officer, and Eleni Perraki, Senior Product Manager. The structure of the outline of the presentation is the following. We will have a 45 minutes presentation followed by a Q&A session. We aim for at least 15 minutes questions. And I recommend to write the questions you might have in the Q&A session of the chat, either during the presentation or afterwards. And without further ado, I leave the floor to Peter.
Yeah, Gianni, thank you, and welcome everybody to this webinar. I'm happy to walk you through our experience with Gaucher disease and show you also solutions we find for diagnosing, but also monitoring people with molecular treatment. And I also want to say, of course, hello to Eleni, the co-speaker. Eleni, maybe you also.
Welcome, everyone. Thank you very much for joining the webinar today.
And so with that, we can really take you on the tour. So Gianni said we will introduce Gaucher disease, which is, I think, not only one of the first metabolic disorders that received molecular treatment, but also an intriguingly disorder where the variability of phenotypes, treatment response, but as well new insights into mechanisms still make research and development a very, very active field. Centogene has developed diagnostic solutions for Gaucher disease, which I will show you in the second part, and then also Eleni will go in detail with a multiomic integration, which is, I think, a very good tool to monitor both the genetics and the biochemical phenotype in one go. Third, we have applied these tools in an observational study that also shows treatment response and severity of disease, which is the Lyso study that will then be the block before we have time for your questions and our answers. So we start with the background of Gaucher disease, which is a genetic disorder, an autosomal recessive genetic disorders, which means we have biallelic mutations in the GBA1 gene on chromosome 1, and the variants there cause a loss of GCase activity with the enzyme activity that represents the pathway structure that happens on this disorder. GCase, or glucocerebrosidase, is needed to break down fatty chemicals, lipids, especially glucocerebroside. And these lipids, they are predominantly seen, especially in the brain and in some peripheral tissues. So we have a huge turnover of these lipids, and this enzyme has to work at good capacities to keep us healthy in our metabolic pathways. If the enzyme is deficient, we get an accumulation of non-cracked complex lipids, and these metabolites start to trigger inflammation, especially in the spleen, in the liver, in the bone marrow, and unfortunately also in the brain, leading to dysfunction of these organs. And we call it a lipid storage disorder, which then leads to symptoms like the hepatomegaly, enlarged liver, enlarged spleen, often due to this accumulation, but also to the inflammatory cells that reside into these organs. When the accumulation happens, we have in the bone marrow especially a problem in producing red blood cells and thrombocytes. So thrombopenia is a very common symptom. And then unfortunately also in the bone pain and even the resorption of tissue in the bone with pathological fractures. We see that patients usually start to onset early, but there is a huge variability in the clinical picture, which has been classified in 3 basic types. Gaucher type 1, where roughly 90% of the patients worldwide suffer from, is a peripheral disease where the brain is spared. We call it also the non-neuropathic form. And in these patients, the symptoms of the liver and the spleen, and of course anemia, and sometimes also the bone crisis are common. Gaucher type 2 and 3, on the other hand, are those with affection of the brain, more severe, often also earlier and more dramatic progressing, where besides the symptoms of liver and spleen and the periphery, also progressive brain damage is seen, especially with seizures, with spasticity, and eye movement disorders or rigidity in Gaucher type 3, the so-called acute infantile neuropathic form. And then we have a little bit less pronounced the type 3 disorder with chronic brain disease, neurodegeneration, and on top all the skeletal, hematological, and metabolic disturbances. So Centogene over the last 15 years has diagnosed more than 2,500 of these patients. And the reason for the urge for diagnosis was, as I already mentioned, that especially the peripheral form, Gaucher type 1, can be reverted by molecular therapy, which is enzyme replacement therapy with massive benefit for the affected patients. This therapy was brought to the patient in 1991, so we look into more than 30 years of experience with treatment. And this enzyme replacement, first extracted from placental tissue, later with recombinant enzymes, has done wonderful for the peripheral symptoms in many patients. They regained a high quality of life, were dependent on the infusions every 2 weeks, but apart from that, really could step out the circle of the deterioration of their metabolic systems and secondary complications. Unfortunately, Gaucher type 2 and 3, the neuronal forms did not respond well to enzyme replacement with a simple reason. The recombinant enzyme, the protein that is given intravenously, does not pass the blood-brain barrier, and so any deficiency of the enzyme in neural cells cannot be replenished by enzyme replacement. Currently, there is 2 treatments approved in Germany, and there is also for patients that develop antibodies or are not eligible for enzyme replacement therapy, also an option to reduce the substrates of the pathway, thereby reducing the pressure in the system and also bringing clinical benefit to patients that are otherwise not benefiting from enzyme replacement. They are given orally, which is a big advantage, but the effectiveness of treatment is higher in enzyme replacement. What we see on the clinical side is that really almost all symptoms that we see with the disorder, the blood count, the organ size, the bone marrow, but as well the growth normalize or at least massively improve, as does kind of the development of the children and the quality of life. Gaucher disease is also, of course, as a metabolic disorder, monitored with biomarkers, and in clinical practice, there have been early markers like chitotriosidase, shown here in the middle, that was almost discovered at the beginning of the therapeutic era. Chitotriosidase is produced in the Gaucher cells, which is kind of the pathognomonic finding in bone marrow if you do bone marrow aspiration. But while the sensitivity is quite good, with more than 90% being elevated in Gaucher patients, the specificity is reduced because we see sometimes even healthy ones having high levels of chitotriosidase with no relation to Gaucher disease. CCL18, a marker shown on the right, is chemokine that is produced by macrophages that goes also up, but is no longer in use because neither sensitivity nor specificity qualified this marker for clinical use. What we work with and very successfully is on the left glucosylsphingosine, which we have brought to clinical testing in 2018, which is a metabolic product of glucosylceramide. And it's so useful for diagnosis and monitor treatment because the sensitivity and specificity for Gaucher disease is close to 100%. We will show this in more detail on the next slide, where you can see that Gaucher patients, and this is publications that have been done by us, but also by others, Gaucher patients show a great reduction of this biomarker from the hundreds into the few dozens over weeks and months and even years, with a stabilization in a steady state where they get regular treatment and then as well keep their biomarker levels low, showing also that the treatment is efficient in the pathway where it should end up. And we see in different treatment paradigms that also you can kind of keep a stable state of disease with limited symptoms over many, many weeks, months, and years. So we have realized that lyso-Gb1, which is glucosylsphingosine, is really a state and trait biomarker that has big value in helping diagnosing and monitoring patients with Gaucher disease. I just have another slide that wraps up also the diagnostic discrimination of these markers. And here you can see that we have for Gaucher disease patients always markers of lyso-Gb1 above 10 nanogram per milliliter blood. And in severely affected, it goes into hundreds or even above 1,000. While in heterozygous healthy carriers, we have usually values above around 5 nanograms. And the separation of the two groups, even for carriers, is perfect. Wild type healthy controls are even lower than the carriers. And so that's illustrating the perfect sensitivity and specificity of that test. What you can see with the marker is, for example, here a case where we did a prenatal diagnosis for a child to be expected to have Gaucher disease, and then got during the first month of life repeatedly marker measurements that showed that it gradually increased with a severe variant, as you can see, a homozygous N201S. And then after 4.5 months, when treatment was started, there was a modification, but we don't have any further follow-up data, but see that together with the clinical picture in this newborn that was clearly affected, we also could see that the burden with this biomarker built up over time. In contrary, when a patient diagnosed at age 15 with Gaucher type 1 phenotype received enzyme replacement therapy, we could see with an undulation of the markers that the values continuously went down in the low 20 to 40s, with stabilization over quite a long time, which gives also the physicians a confidence that the efforts he's take to reduce the metabolic problems and the effect of that treatment are in balance and really benefiting the patient. Now, this is the utility in developing the markers, but we also thought to have more integrated products, and that's where Eleni now will take you for the next couple of slides.
Thank you, Peter. And so I will talk a little bit about the concept of multiomic-based approaches in clinical diagnostics. So when we're talking about single-omic approach, is basically genetics. So it's studying one type of molecule per test. However, this sometimes may yield not conclusive results, and this may result in delaying the diagnostic journey and also causing inconvenience to the patient and causing the necessity of additional specialists to be involved in order to be able to reach a diagnosis. On the other hand, when we're talking about multiomic approaches in clinical diagnostics, this means that we are integrating different type of data sets, so different type of molecule analysis in one test. And these different data sets are analyzed as an integrated comprehensive data set. And this can help give more insights into the disease and the diagnosis, and also giving a better understanding of the whole clinical picture. So a multiomic testing then can provide an earlier diagnosis and also help towards optimized treatment strategies. So multiomic approaches then is, let's say, a more holistic clinical picture. How can that be? So what can be integrated when we're talking about multiomic data sets? So we have genetics, of course, and then additional dimensions in a multiomic approach can be transcriptomics, so RNA sequencing. It can be proteomics, metabolomics. It can be epigenetics. And then these different data sets are analyzed as a comprehensive data set, and it can provide insights into a more precise diagnosis, into improved prognosis for the patient and optimized treatment strategies. It can help understanding the disease better, so getting insight into disease subtype, discovering new disease gene associations, and also on the side of research, it can also provide information about new treatment targets and biomarker discovery. Next slide, please. So as Peter mentioned, in our lab, we have a lot of experience in diagnosing Gaucher disease patients. And we have diagnosed around more than 2,500 patients. So one of the observations that we have made is that when on top of the genomic data, we are also adding an additional multiomics layer, which is the biomarker Lyso GB1. This can increase the diagnostic yield in Gaucher disease of up to 25%. And this is particularly the case for Gaucher disease because the gene that is associated with Gaucher disease, GBA1, is a complex gene that it has a pseudogene, and a lot of the times the genetic results might be complex to interpret or unclear. And in these cases, having the additional information of the biomarker accumulation can help reach a diagnosis for these patients. So the next layer of multiomic analysis that we have included in our multiomic approaches is RNA sequencing. And what can the transcriptome tell us? So first of all, we can receive information regarding the gene expression. So genetic variation can alter gene expression levels, and this can be visible using RNA sequencing. Another piece of information that we can receive and we are particularly interested in is splicing events. So genetic changes can cause alternative splicing and can produce alternatively spliced mRNA products. So we are using this. So this is an information on the functional effect of variants, which can be very useful towards variant classification of variants of unknown significance, which then in turn can help stratify patients that we wouldn't have been able to diagnose otherwise. Next slide, please. So our experience with multiomic approaches in disease diagnosis is that when we are able to integrate genomic, biochemical, and transcriptomic data in one test, this can help accelerate the diagnosis of the patient. It can have better disease prognosis for the patient, and it can also enable personalized treatment approaches. So the different multiomic tests that we have vary from a panel, our centabolic MOX, which is an NGS panel, which is focused on inherited metabolic diseases, and the multiomic dimension that is added here on top of genetics is biochemistry. And we have actually a study published of the first approximately 4,000 patients using this test that can give you information about how advantageous it can be, this sort of multiomic analysis in this type of group of diseases. And then other multiomic analysis that we are using on a routine basis is centoxome and centogenome multiomic approaches, so exome sequencing and genome sequencing. And as a multiomic dimension, we are adding biochemistry and also RNA sequencing analysis. And this can be particularly interesting in terms of, and useful in terms of genome sequencing due to the scarcity of information in public databases of non-coding variants. And one important thing I would like to mention here is that these tests are all performed from the same starting material. So we are using our blood filter card, CentoCard, as the receptacle that we are extracting then the material that all the different multiomic analyses are performed. Next slide, please. Here we have a clinical case that I will talk to you about. So this was a 15-year-old male patient that was presenting with intermittent abdominal pain. There was no family history of genetic diseases. The parents were consanguineous, and gynecological examination showed no conclusive results. Physical examination suggested hepatomegaly. We performed centometabolic MOX analysis, and we detected a homozygous missense pathogenic variant in GBA1 gene, and biochemical testing showed increased levels of the Lyso GB1 biomarker and also decreased levels of the enzymatic activity of beta glucocerebrosidase. So with centometabolic MOX, we were able to confirm the diagnosis of Gaucher disease, and biochemical results confirmed the pathogenicity of the variant, and we were able to establish a precise diagnosis. Next slide, please. And here you can see the results of this of the multiomic analysis. So you can see on the first level the genetic testing. So you see the gene and the variant and its classification, and then on the multiomic level, so on the biochemistry level, you can see the lyso-Gb1, the accumulation of lyso-Gb1 that was in pathologic levels, and also the decreased enzymatic activity of beta glucocerebrosidase in this patient. So here you can see how a multiomics approach can give a holistic picture into the clinical presentation of this patient. Peter, over to you.
Thanks, Eleni. And I just add using the tools and these clinical tests within an observational study. We initiated a study where we tried to find naive patients with Gaucher disease that have not yet received treatment, and use the biomarker and our tools to understand as well disease severity correlated with genotypes or with clinical picture. And in the next step, when treatment was started, to show that the biomarkers also always paralleled the clinical efficacy of the enzyme replacement therapy. Can imagine that finding treatment-naive patients is not so easy in well-developed countries, and so we had many of these countries in North Africa, Egypt, Pakistan, India to find also patients that were untapped, and you can see in these sites we went for a total of 160 treatment-naive Gaucher disease patients, and could see at baseline without treatment that the markers correlate with the clinical picture. And of course, this should also help to see how far treatment effects go in similar cohorts than under treatment. So really having a baseline there. As Eleni said, this was all done using the Cento card. So we received just dried material, which made it very easy also to have logistics from countries where it takes sometimes a couple of days longer with high data and then as well interpretation quality. This is just to illustrate how colorful a Gaucher clinic can look with it in Pakistan, in Lahore. And you see those children that showed up to get diagnosed. Not all of those have Gaucher disease, so it was also to establish a diagnosis. But we have been meeting with many, not only patients, but also the parents, and it could also help them to understand better what is a genetic test and what we are doing when we bring a diagnosis and then maybe eventually also treatment. And the study design in this observational was a multi-center study which went for untreated Gaucher disease patients to demonstrate the correlation and the predictive value of lyso-Gb1 with the clinical severity. And you can see it was kind of for patients above 6 months, which first had to receive a genetic diagnosis and no prior treatment in the last 24 months. The workflow was straightforward. So blood was taken, dried, sent to the central lab. We did kind of the technical analysis for the biomarker and confirmed the genotypes. And there was also severity scores that were used to assess the type of disease and the accompanying symptoms. And we have published that, and you can see kind of the reference here on the slide bottom right. But the main finding was, in fact, we could correlate the biomarker with the severity score that had been developed for Gaucher disease, the GD-DS3. Not super high correlation, but showing that the more severe the clinical picture, also the higher the biomarker levels were with an R of 0.6. And we could also show that in type 1, there were subpopulations that had lower or higher severity scores that also correlated once again with the lyso-Gb1 levels, with the more severe having significantly higher levels than the mild ones, and also the non-classified variants, which is a mix of type 1 and type 3, genotypes had on average the highest levels, which means we have quite consistent results on the representation of biomarker levels for these cohorts with the clinical picture and also with the identified genotypes. To wrapping that up, I think we have an excellent tool set to take care on a not so rare metabolic disorder. We see that Gaucher disease is among the top 5 we diagnose in the metabolic space, even with a broader indication or search. And we can compile our knowledge of the last 15 years dealing with Gaucher disease, but also related lysosomal or metabolic disorders like Fabry and Pompe, with a well-defined biomarker portfolio menu that helps to differentiate lysosomal storage disorders, where lyso-Gb1 is a prime example of how good a biomarker can help in clinical practice, but also in clinical development. And so we support early in clinical research, but also in access to the treatment by finding patients and also understanding better also ethnicity differences and clinical picture differences in cohorts all around the world. We also used the knowledge around this biomarker to go early preclinical in cellular models and in a cell model with macrophages derived from patients. We were able to even find new small compounds that modified lyso-Gb1, reduced it in Gaucher cells, being excellent candidates, maybe even to close some unmet needs gaps in Gaucher disease for the bone disease and for the neuropathic forms of Gaucher disease. And in this and similar collaborations, we also, of course, showcase how our research teams with their capabilities in cellular models, our biomarker team with their understanding of how to measure disease relevant small molecules or markers, and of course, partners and patients come together. Now, the whole pharma team, and that's part of this presentation as well, is also helping with clinical trial setup and the elements we can really provide, especially in lysosomals and Gaucher disease, is monitoring treatment response with easy logistics and highly sensitive and specific markers backed on an accredited laboratory and established fast turnaround workflows. So I think this is also an opportunity to reach out to our pharma team and then discuss needs where our expertise is not only the operational, but also the knowledge we gain by collecting the diagnostic and the pharma partnership cases in the biodata bank, where now, as Eleni said, more than 2,500 patients have been collected. And we can slice and dice according to genotypes, to phenotypes, to ethnicities, to geographic regions, and help to understand where to find patients when you look in more detail for maybe even phenotypic subtypes. Last but not least, this is kind of the value to shorten time with patient identification in the biodata bank, but also to go deep on our biochemical biomarker capabilities, discover new biomarkers that are disease-related, or use the existing biomarkers to measure severity in models to maybe clarify small molecule mode of actions in pathways, and there were also de-risk early clinical programs. With that, maybe I should, in the end, just say what we learned most in Gaucher disease is that It's a rare recessive metabolic disorder, but since 20 years, it's also known that carriership for GBA variants is a risk factor for Parkinson's, which might affect even more patients than the Gaucher patients. They need different research. They need different models to understand what is the pathophysiology. But Centogene and the teams here have collected information to provide tools also to do research on the GBA1 Parkinson side. And with that, I think we close the presentation and go back to Gianni. And
in that case, let me wrap it up. So thank you, Peter, thank you, Eleni, and thanks to all the attendees. We hope you find it instructive. And see you for the next webinar, probably.
Yeah, thanks a lot.
Thank you, everyone.
Yeah.